Our group here has interest in the metabolism of a class of compounds that are called quinones. They are at the core of the energy production machinery of all cells.
In this lab, we work on model organisms, so it's like plants and cyanobacteria. If we can figure out in these organisms how they work at the molecular level, then we can then take that and apply it to organisms on a broader scale. So it's kind of like putting together that puzzle and then applying that.
The importance behind our research is that these compounds are vital for respiration and photosynthesis. Understanding how these compounds are made, adds to the scientific knowledge, and then also can add, that knowledge can then be applied to that of engineering plants for either increase in nutritional content or with that of ubiquinone, showing an increase in the content of ubiquinone in a plant has conferred stress resistance, leading to more plants that can tolerate more stress or end up having higher yield.
It was a very basic science project which was initiated by looking at some enzymes involved in electron transport. And he was interested in the question as to why things evolve the way they have. But the end result was he discovered an enzyme and an inhibitor for that enzyme, which might be a very potent herbicide, or at least analogs of that.
We discovered that we could manipulate this metabolism to manufacture some new herbicide. There is a desperate need in the industry for new herbicides. It's a little bit like antibiotics. Plants have developed resistance against herbicides, and the industry really need new molecules and herbicides with new mode of action, so they could rotate them on their field to avoid the emergence of weeds that become resistant to all kinds of herbicides.
So I chose it because it was a unexpected result from this very basic science project, but it might end up with something that has a great deal of practical application.